Hey, Professor Leslie. Sorry we didn't get the chance to do this in person, but I'll just do the best that I can with this video type thing. Alright, so I did the Institute for Genomics and Systems Biology, or the IGSB, which is a lab in the University of Chicago. It's the one I worked at over the summer. The presentation is called War on Cancer because, like Pavlov's physiological factory, I think that the IGSB has a battle plan most similar to the Army or some military engagement. Looking at the people and divisions of IGSB, there are two individual leaders, the Richard Jones and Kevin White labs. The Richard Jones lab is smaller. On average, they have about 10 people. The Kevin White lab has about 20, 25 people. I mean, it varies depending on the time, how many lab techs and such, but those are general numbers. And then in the Jones lab, which is what I'll be going into more detail, there are four sort of divisions, as I've labeled them: protein signaling, lab technicians, physicians, and other support, like even other people outside the lab, or statisticians, and then geneticists. And then looking at Richard Jones himself, I gave him Major General rank, which in the Army is a 2-star general, command 10 to 20,000, and all that really means is capable of a fairly large impact operation, and then capable of fully independent field operations. So, not necessarily doesn't answer to anyone, but can make very real decisions and makes them all the time. All right, the real Richard Jones, everyone calls him Rich. He's an assistant professor at the University of Chicago, so he does not have tenure, which gets interesting. His main duty for the lab is to get money. He writes research grants all the time, but he also ensures quality research through everyone else's project. And his most interesting comparison with history, I believe, is Ivan Pavlov. And his differences from Pavlov are he allowed choice in topics. So, people would sit down for lunch, they'd talk, they'd throw around ideas, they'd talk about sniping projects from one another, giving them, oh, I'll give you a fourth author, as a joke. And he does not care when you do work, because he himself a lot of times did not roll in until around 3 o'clock in the afternoon. It did not roll out until about 5 in the morning. So it was very interesting how that worked. He worked very strange hours. People were always curious as to how he could possibly have a life outside of it. But the similarities to Pavlov is that he conducted lab-wide meetings, like Pavlov did, once every 2 weeks or so. And then his one-on-one meetings were absolutely crucial. Had those every week. And then I had the pleasure of sitting in on one. He really tears into you. The grad student that I was working with, the method he chose was a good method, but he didn't do it in quite the right way. So he really got torn into for that decision. Moving on to Lieutenant General Kevin White, as I've labeled him. Lieutenant General in the Army, leads an Army Corps, around 60,000 soldiers, so capable of a large impact. And then another thing is they're in the armed forces longer, so they achieve this rank. So he has been active longer than Rich has. Similarities to his lab position: he does lead a larger lab. He has greater resources, and we always talk about the -70s, which are the really cold freezers. The Jones lab has 2, and his lab has 7, and they have extra space in theirs, and the Jones lab is just busting open. We had to go and ask a friend in the White lab to borrow their space and stuff like that. He is a full professor. He has tenure, so he's much more chilled out than Rich is. Looking at the Jones lab, sort of combat divisions that I talked about earlier, there's protein signaling in cancer cells, and they work by identifying the problems and attacking them. So they identify, what does this cell have cancer? Does it not? How can we attack it? How can we fix it? Lab techs, they help to fight with brute forces, sort of privates on the front lines, charging headstrong in a battle. I was more that brute than anything else, although I don't fit perfectly into it. Physicians and other help, so they help getting clearance. If you're dealing with human tissue, you need physician clearance. You need physicians to get the tissue cultures, to do all that stuff. And then other help, we had a statistician collaborate, we had a few other labs collaborate, and then also looking at a genetic influence. So they locate the source, attack it in different ways, sort of like, how can we prevent this problem from happening? Or how it's a softer way I think. It's not as, charge into battle. It's more of a peaceful negotiation way. Still fits into the Army, but still it's less about dealing with the problem. It's more figuring out the best way to deal with the problem, which will make more sense later. Looking at protein signaling mechanisms, how they work is they look to see if cancer is present. That's one of the things they do. So they look for certain proteins that are inserted in the membrane that show that it's growing uncontrollably. They look at ways to prevent this further growth, or keeping it from spreading. A lot of things they deal with cancerous tissue, so they actually use some cells that are cancerous, and they deal with it. And they also use proteins, which they make with the peptide synthesizer. A lot of times they use mass spec to identify a protein, to identify their own proteins that they made. But a lot of it is just dealing with, we found cancer, how can we make it go away? How can we stop it? Lab techs are hired hands, sometimes undergraduates at University of Chicago, because this lab is at the University of Chicago. A true lab tech gets no choice in projects. They get assigned to somebody that needs help, and they do the brute work. They can, though, get their name on a paper, usually towards the rear end, which, you know what, I'll take. They're helpful in the execution of experiments, but not so much in the analysis. In the execution, they are very good. They work tirelessly, as I did, for long hours, and sort of the work that you don't want to do, you can have them do. I mean, it's kind of cruel to do that, but you got to get work done somehow. My personal story, because the role I took was more being a lab tech. The micro-western analysis, one of the techniques we did was a micro-western, which I'm sure you know what a western is. It's looking at proteins. The micro-western is something that the Jones Lab actually invented, which is 96 times more throughput than a western. So I had to deal with a lot of data, and it was analyzing it by scanning. We'd print it onto a gel, electrophoresis it, bring it onto some paper, stain it with a primary antibody, secondary antibody, the regular steps of a western, and then looking at it and determining how bright it was through a computer. And a lot of that was dragging a circle over a dot. And how I'd have to sit there and drag circles over dots, and it's 96 times more throughput. We had 76 individuals, 4 replications of each individual, and we stained for 15 different proteins on any given time that we printed. So I had a lot of dots to work with. I was there for 3 full days, 9 to 5, walked into the lab, put down my bag, went to the room, circles over dots, circles over dots, circles over dots, the entire time I was there for 3 full days. It can be very, very long work, but it is very helpful in getting it done, and I like to think that I helped him advance his project a little bit while I was there. Physicians and outside help. Physicians, they can get tissue samples from patients, as I remember. They often help through collaboration. They just talk. A lot of times people have meetings with a physician, and some of them do wet lab. There are also some MD PhDs, which kind of count, but some just MDs will be doing wet lab. There is an MD lab, and I only had one run-in with them. Apparently they all wear lab coats, whereas this lab did not, because lab coats are disgusting and filled with dirt, and you wear them every time, so they get even dirtier. At least that was the thought, but that was the only run-in I had with the physician's lab. And then other outsider help, such as statisticians. They come. I met one at one of the meetings that we had. They help with the analysis of data, and they're very useful in determining if the data is meaningful after you get it done. Geneticists in the lab look to determine the impact of single nucleotide polymorphisms. They're helpful in determining why people respond to certain cancer treatments. They use human cells, microwesterns, RNA sequencing methods, and they collaborate with the white lab, because the white lab had a lot more geneticists than we did. But SNPs, I'll elaborate that on. I realize I just skimmed over that a little bit. If you don't know what they are, single nucleotide polymorphism is what it stands for. It's basically a single letter change in DNA. In that strand of 3 billion, that seems, I don't know what the actual number of letters in the DNA is. I probably should remember that, but fortunately I don't. Single nucleotide polymorphism is a one-letter switch. Determining why certain people respond to certain cancer treatments. One of the meetings I sat in on, they were actually just looking at data for African individuals. They had been given chemo, and they had also had their DNA sequenced, and they looked to find that one-letter difference that made a difference whether it was successful or unsuccessful in eradicating the cancer. So I thought that was very interesting in how they lined that up and tried to find that one letter that made a difference. But that's a lot of what geneticists do. So then looking at my grad student in particular, I gave him the title of, or the rank of captain, Ron Haas. Duties in the Army would be commanding 62 to 190 soldiers, assisted but still somewhat free, and helps in combat training centers. So the similarities to the actual lab, has a small help team of lab techs, if any at all. A lot of times he didn't, but now since he started his project, he's had 1 to 2 the entire time. He still reports to the lab director, and it was an unusual scenario with me. I didn't get paid money, I just got paid knowledge, I guess, which I was more than happy with, because he really taught me a lot about what he was doing and helped me understand it, and that took up a lot of his time, but I still appreciate it. And that, help in combat training centers, I mean, pretty much helped train me for my future, hopefully my future career in research. And then comparison to history, I think he's most similar to the mentor chain, because his project is fundamental and risky. It could definitely have a high payoff, which I'll go into later. And then looking at why I called it a war on cancer, because I realize I haven't really touched on it yet, and I'm 13 minutes into this. As far as how the people go, I think that it works, but the real reason that I call it a battle plan is because of how they attack things. So I will go into their philosophy, the similarities with that philosophy to real battle, the methods of attack, and the resources that they can use. And then also in war is hell, as it's been said. And obviously lab research is not hell, but it can still be very stressful, and they have a similar way of just sort of kicking back, staying sane the entire time through. So the Jones Lab philosophy, this is taken straight from the website, pictures included, straight from the website. There's a little tab that you can go to that has just this. So when approaching a difficult problem, perform the following actions to obtain the best tools for the task at hand. Test your tool on a small, scalable, tractable problem. Pick an extremely challenging but achievable goal, dive in and make necessary corrections in mid-flight. Enjoy the ride and enjoy the destination. So that part, not so much war-like, but the rest of it, I think it is. I think it's very aggressive. So the Normandy invasion is the military operation that best exemplifies the Jones Lab philosophy. And the highlights of the storming of the beaches was they chose the best time to strike. And it wasn't the time when they could get closest to the actual place. It was the time when the tide was lowest, so they had to go further on land, which was the least expected time, but ended up being the most successful. It chose the right technology to get the job done. They had to drop them off in the water and have them run up. They gave them what they needed to give them, and they got it done, and they threw everything they could. It wasn't, it's not a, strolling of the beaches, it's a storming. They went at it with full force, with everything they had. So the Ron Haas thesis project deals with the central dogma of biology that DNA leads to RNA, which makes proteins, which do things. So his individual project was looking at RNA to protein, and looking at the levels of RNA to the levels of protein that can be found in any individual cell. So it's a fundamental project. It deals with the central dogma of biology. It's never been looked at before. So it's very risky, except for one thing, which was a small study, which said it there was no correlation. So he chose a different protein, which I think was a great idea. And if it's successful, it can start a chain of high impact research because it has a lot of potential with literally everything, literally all disease. I think it exemplifies many characteristics of D-Day. He chose the best time he could to start research. And I sort of think the best time is sort of the best protein, because in D-Day they didn't choose the time everyone thought they would. They chose a sort of roundabout way that would be most successful. And I think with the protein he chose, transcription factors, there's a very strong similarity in that. And then he used the most advanced tech available, the microwestern, used the biggest resources available, HapMap, which, if you don't know, is sort of a worldwide genome sequencing project. And then he has been working on it relentlessly. I've recently talked to him. He's still really just cranking away at the lab bench. So highlighting the similarities, the paper came out with the inconclusive results right before his. He figured it was the best time to gain publicity for the lab as well. That was one consideration. He thought, he'd make a splash. Choosing the technology and resources, he chose the microwestern, HapMap, 96 more times throughput than any other tech that's been thus used in the HapMap project, which, is very helpful in identifying SNPs and such. And then keeping after it, as I said, he's still working on it, and he has consistently had 1 to 2 lab techs helping out. Right now he has 2. And then for the last part, sort of staying sane in the war, because research takes a very long time. He's still working on it, and I was working on it over the summer, and he's still nowhere near being absolutely finished. One thing they do is they have conference room ping pong. They take the tables and they set up a net. They just take ping pong breaks throughout the day. And then every Friday at 5, they have happy hour. They take beer out of the cold room, go into the ping pong room, play some ping pong, and drink some beer. And then, sort of as I was leaving, there was, for the happy hour, it was like a happy hour / going away party. So I brought in some cupcakes, which is very awkward walking through downtown Chicago with a huge tray of cupcakes, but worth it. I made some DNA, double-stranded, RNA single-stranded, and protein, which probably tasted nasty because that's gum that makes the alpha helices, but oh well. And then another contribution, my only real contribution, the mitosis cupcakes, which hopefully I'll set it up so you can sort of see what it's like, but interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and then a new cell. But in summarizing it, the most important thing is that the Jones lab is very small. There were only 10 people there when I was there. There's only 10 people there on average, and they're still putting forth this very impactful research. The microwestern alone, which was published in Nature Methods, which not as exciting as Nature, but still that's a big step forward in looking at protein function. The fact that you can look at protein at a much greater rate with near the same accuracy. And it made me hopeful, because if every lab is doing as much work as the Jones lab, there's no surprise that we're learning so much about everything. It's such a fast rate, because the research being done there the quality was very high, and it made me very confident that cancer's days are numbered. So then I have my sources, and then thank you for watching. Thank you for listening. It's a good class semester. I hope to cross paths with classes in the future with you. Thank you, Professor Leslie.